take a seat. We're about ready to get started. Thank you, everyone, so much for being here. It's really amazing to see everyone here in person. Before we dive into the data blitz, Alex and I just wanted to take a minute to welcome everyone and set the stage. A couple of years ago, Alex and I kind of independently had a thought that wouldn't it be nice to get us all together, and we took it to Isa, who took it to Richie, and voila, here we are. Yeah.
It's honestly mainly center staff and Isa's team, and folks like the Discovery Center team that made all of this come together so smoothly.
Yeah. While we're on that point, huge thank you to Isa, Heather, Nikki, Taylor, people I don't even know who've been behind the scenes working on all of this to make this weekend a really special weekend for all of us. They've done a lot of work, so thank you so much. All right, well, looking around this room, it's hard not to feel the size and the impact of the legacy you built, Richie. People are here from around the world, from across decades. There's people who couldn't make it here but are here in spirit. And we're all here to share with each other, see old friends, make new friends, and express our gratitude for our common connection, which is Richie. I'm going to start with a quick personal anecdote, kind of my origin story, I guess, if you will. I had been an undergrad research assistant in the lab for a couple semesters, and my boss, Andrea Strauss, was leaving for grad school. And I remember Bonnie said, "You should apply for the job." And that totally blew my mind, because at the time, all of the staff in the lab were very experienced, more senior people. There were no post-bac staff in the lab. And I had just graduated. I came from this tiny blip of a town in Wisconsin, and I just could not picture myself in the company of these people. But Bonnie's like, "No, do it." And I think you might have had to convince Richie, because I think he might have had the same uncertainties I did about this. I remember when you called me to offer me the job at home, and you said, "Hi, Chris, I think we want to hire you." And my first reaction was, "Okay, maybe call when you're sure." My second reaction was, "Yes." And my third reaction was, "Chris, don't F this up." And I guess I didn't, because I'm here. So to the many post-bac staff that came after me, you're welcome. Anyway, Richie, I'm forever grateful to you for taking a chance on me. It was during that full-time position in the lab that I realized being a scientist and academic was my dream, and you gave me a chance to pursue that dream and the opportunities to really make it happen. So we all have different stories here, but one common connection is that at some point, Richie took a chance on all of us and gave us these unbelievable opportunities to flourish. So thank you so much for your past generosity in forging our careers. I'm not going to cry. And your current generosity in hosting us for this really wonderful weekend. Thank you, everyone, for being here. I'll pass it off to Alex, maybe, for nuts and bolts?
No, you do it.
Okay. All right. Speakers, you've been warned about the time 17 times. Please stay on time, because we have to be out of this room at 4:30. I've been threatened with this, so stay on time. I will sit right here. I have a sign that says you have 2 minutes left, 1 minute left to get you to 5 minutes. Drew has a backup squawker that he will squawk at 5 minutes and double squawk at 6 minutes, at which case I bust out my mom voice and pull you off the stage. So please stay on time. And with that, I'd like to introduce our MCs, Drew Fox and Regina Lapate. Yay. Thank you. And they're going to take it from here. Welcome, everyone.
All right. I will try not to waste any time here, though I don't know how do we get the slides up, guys?
There should be a...
There we go.
What? You're in the way. There they are.
Hey, there we go.
Slides up.
Okay. All right. Huzzah!
Huzzah!
Huzzah! Thank you all for coming. We will try to, as moderators, use as little of your time as possible so that our speakers can regale you with their past and present accomplishments. First off, we're going to introduce Leandro Chernicoff, who was a graduate student in the lab from 2016 to 2023. Come on up, get started. Let's not waste any time. Don't wait for your introduction. Bust up here. Let's do it.
Okay, hi everyone. It's time. When are we starting with the time?
Right now.
Okay, good. Is it Mexican time? No. So, thank you so much. I'm really happy that I'm the first one, especially because then I can enjoy watching your. Otherwise, I would be too nervous thinking about my presentation. So, basically, when I started my PhD, and a little bit before I started my PhD, I asked Richie, what should we be doing, and what should I be doing? And we work at Atentamente, this Mexican non-profit, and how can we help? And he said, basically, as he said yesterday, we know this works, but the issue is, can we make it at scale, and can we look at things that are policy relevant? It seems that we've had so much data already that's showing that it's working, but for some reason, people are not listening. Maybe we need to look at all these sort of more distal outcomes that will make people put attention. So basically, this is what we've been trying to do for the past years. And basically, I will tell you a little bit about these two large RCTs that we did in Mexico. When Richie said, do something big, this is what we tried to do. So we did these two large RCTs, one in education, one with healthcare providers, one with preschool teachers in the Sinaloa state, and then another one with healthcare providers in 7 states in Mexico City. And basically what we did is we looked first if there were changes in proximal and intermediate outcomes, and then look if we saw changes in the proximal intermediate outcomes, then looking at the distal outcomes. So very briefly, in the Sinaloa study, we found that there were significant and positive effects on all the things that we were looking, educator socio-emotional competencies and well-being. And not only that, we also found that there were improvements in students, the children, socio-emotional competencies. And that gave us some sense that, okay, this intervention is doing good things for the students and for the teachers, but we did not know if that had an impact on grades, especially standardized grades. So we're working in many states in Mexico, and another place where we were working is the state of Nuevo León, which probably has the best evaluation system in the whole country. And because we have been working with the Ministry of Education, we had access to the data for these standardized measures, and we were able to develop this quasi-experimental design in which we could look at the standardized grades, math grades of the kids in 3rd grade, kids that have received, teachers have received the intervention versus a matched control group. And we saw that there was improvement in standardized math results. And I think the nicest thing about this is that it's for kids with in the lower SES range, those that need it the most. And we're doing a lot more. The sample size here is around 20,000, but now we're with Matt, wherever he is, we're doing something with about 50,000 to 100,000 kids. And the other thing I wanted to briefly tell you a little bit about is what we did with healthcare providers. This is something Matt took the lead. And also what we found is that for the proximal outcomes, meaning the healthcare providers' competencies, awareness, connection, insight, and purpose, and their well-being and distress, we were seeing good results, positive results that also lasted 6 months after the intervention. And then we asked the question, well, if this is, if we're seeing this, what about the more distal outcomes, productivity outcomes? And we work with this fantastic Mexican economist, Fernanda Marquez Padilla, is a very good friend, very bright person. And she took that data and then matched this data with administered data that we had from hospitals and healthcare centers all over these places where we're working. And what we found basically is that after the intervention, the productivity rose from somewhere between 8 to 11%. Why is this 8 to 11%? We're still analyzing a few things. And the important thing about this is that there were statistically significant improvements in healthcare provider productivity, but at the same time, in general, it had no impact on the quality of care. because you say, well, they're taking care of more people, but then the quality of care is decreasing. So on average, at all, there was no changes, for example, in mortality rates. And something that I thought Matt was going to present, but we all deferred to and I respect Matt dearly. I thought he was going to present that. But then the other thing that we see is that in the ERs, there's a statistically significant decrease in mortality rates, which is something quite interesting. So that's all. Am I good in time? I can dance or something. So I'm good. Thank you. And thank you, Richie. Thank you.
That was beautiful. All right. Please welcome our next speaker, Anna Finley, who was a Davidson Lab postdoc from 2019 to 2024. Yay!
Hey! Thank you all. It's wonderful to be back in Madison and see so many familiar faces and just see all the joy of everyone coming together. So this has been really fantastic. As you may have noticed, my tenure in the Davidson Lab overlapped with the pandemic, and so I don't actually have a lot of pictures in the office with Richie, but I do have fond memories, and I think some other folks may as well, of me taking Zoom meetings from my backyard in Madison with my chickens or my new puppy, and making do with what we had to do in the pandemic. But before the pandemic started, the first time I met Richie, I think, is really emblematic of kind of what Chris has mentioned before and kind of alluded to with Richie taking a chance on all of us and opening doors for us. So I was supposed to show up right at 9 a.m. sharp to fill out all the employee onboarding paperwork, and little did I know that the doors would be locked, right? CHM has automatically locking doors. And I think this was before the package button had been installed and for people to ring for the front desk. So I show up and I try the door and doesn't open. I'm like, oh no, what do I do now? I don't have anyone's phone number. I've just moved to town. And I turn around, and walking across the parking lot is Richie. And he so graciously opened the door, warmly greeted me, and helped me get on my way to the right part of the building, which was really fantastic and wonderful. And I think really that warmth and compassion and care that he brought to that interaction, I think really is emblematic of what he's done for all of us in various ways. And, in seen and unseen ways, he's opened doors for all of us to be successful. And I'm very excited that I've just finished my second year at North Dakota State University as an assistant faculty member with my own lab, where hopefully I'll be able to pass on some of that grace and wisdom that Richie has given us. So I wanted to allude to a few things that my lab's up to. Still in my second year, so things are still kind of getting up off the ground. I'm very excited to have a pilot study ongoing right now, actually, looking at the affective neuroscience of loneliness. And so I started some of this work as a postdoc with Richie with my F32 fellowship, and now I'm able to collect some new data looking at EEG neural signatures of emotion and social reward processing in people who report being high and low in loneliness. That data is still being collected, so stay tuned to hear more about that, and I'm happy to talk more about that as well. I now have students of my own, and so Janny Western is my first graduate student, and she's very interested in veterans' well-being, particularly once they return to civilian life. And so we've been starting to explore this using some existing data sets before we start collecting our own data. And so this is from the Military Health and Wellbeing study of almost 1,500 veterans. And here we can see that in this case, military self-stigma, or kind of how people are feeling about their past military service in more of a negative way, is moderating how much feeling like you're socially integrated with your community or contributing to your community socially matters. And so if you have a lot of this negative self-stigma about your service, you end up seeing less of a benefit in well-being from these social indicators, suggesting there's something important going on with how people are kind of perceiving themselves and their identity. I also have a lot of really talented undergraduate students, and so this is Alexa Bed-Dixon, who is an undergraduate student in my lab who is working on an NSF undergrad research experience this past spring. And here we looked at some data my lab has collected, looking at kind of what leads to loneliness. And she found some really interesting findings with authenticity, and in particular, authentic living, where students who feel like they're living an authentic life to themselves report less loneliness. And part of that is through university belonging. And the other part is through self-alienation, or kind of feeling like you don't know yourself. So if you live authentically, you feel like you know yourself more, feel less loneliness, and you feel like you belong more in the university. And so we're, of course, going to follow up on these and many more studies. Janie, in particular, is really interested in looking at how veterans' sense of identity when they return to civilian life impacts their well-being and ways in which they can kind of have that transition go in ways that will help them flourish. And then Alexa is interested in understanding if we can perhaps alleviate some of the isolation our undergraduates are reporting by helping them find ways to live authentically, particularly on campus. And that's just a small taste of what my lab is up to. And once again, I want to thank Richie so much for everything you have done for myself and I think everyone else in this room. So thank you.
All right, so it's my pleasure now to introduce one of our MCs, Drew Fox, who was an undergraduate in Richie's lab between 2001 and 2002, a staff member between 2002 and 2005, and a graduate student between 2005 and 2013. So welcome, Drew. I'll let you.
I should have brought the chicken up here so that I could cut them off before they cut me off. All right, I'm now a professor at UC Davis. My lab is really focused on this sort of general idea that we can alter a specific molecule in select brain circuits to alleviate psychological suffering. We use a bunch of different approaches to this, and today I'm going to tell you about computational models and how I think that we should be using formal models in order to better bridge across levels of analysis, from genes and molecules to cell types, distributed circuits, behavior, and ultimately subjective feeling, which is what we care about. To do this, I have to contextualize some of the work that was done when I was a graduate student, even by somebody who's, I think, somewhere in this room. There he is. Dr. Dan Grupe, working on this sort of idea that uncertainty was related to anxiety, could lead to altered extended amygdala function. This idea has had a huge amount of traction, and the general idea behind this is that when we have a direct threat, our amygdala and our central nucleus of the amygdala respond, and that leads to fear, or an uncertain threat leads to the bed nucleus of the striatum terminalis, which leads to anxiety. I have been wildly critical of this sort of general view, both in my private and public life, and it really stems from this idea of like, what is uncertainty actually? What are we doing? And so we tried to take a peek into this, thinking like, how likely is the threat or the probability? How certain is the outcome, or what we're calling the risk? How certain is my estimate of the probability, which we're calling ambiguity? I should note that these are all terms that are borrowed from behavioral economics. And how uncertainty about when the outcome will occur. And to say, like, when we say uncertainty leads to anxiety, what exactly are we talking about? And first of all, it's not this one. We published that. The timing of a threat can be reduced to the perceived probability accounting for temporal dynamics. But let's push this forward. So now we developed this task. This was done in collaboration with Dr. Alex Shackman, another esteemed alum. What we did is we have a very simple task where we just present a cue that indicates the probability that the participant will get something that is bad. In this case, a shock, a picture, and a sound all together, all at once. But in doing so, we can also look at how certain are we about the outcome, right? If something is 50-50, it's not, it's just uncertain. But if something is 100%, it's not uncertain, it's just bad, right? So which one of these is it? Is it probability, risk, or ambiguity? Well, here are some plots looking at self-reported distress, and I can tell you the pink one looks like the big effect. And in fact, we can compare these and show that, in fact, it is probability more so than risk or ambiguity that is driving self-reported distress. Now, of course, self-report has its problems. We all remember that lecture. Skin conductance also shows the same pattern. And so, okay, cool, but what about brain? Well, it turns out brain also shows that pattern. And this is not due to timing, but Okay, cool. But we want to get at the brain circuits, right? What is driving this? It turns out that theory, the central nucleus of the amygdala and the beta nucleus of the striatum terminalis doing different things, that's not true. So we have now probability, risk, or ambiguity. Well, it's not those, but how could this be, right? And in fact, we can replicate prior work in these data, looking at a 50% threat, an uncertain threat, and show that we do get this dissociation between these neural circuits. How could that be? Well, I think this is it. And this is a bootstrap analysis trying to look for what is the activation function of a particular brain region given varying levels of probability. And we can show that the beta nucleus of the striatum terminalis is more responsive to low probability threats than the central nucleus of the amygdala. So BSD activation is preferentially involved in driving behavior to low probability threats. This model is consistent with experiments in that drove the uncertainty model. We actually lesioned the beta nucleus of the striatum terminalis to test a prediction, but that was cut for time. So this specifically low probability threats, I think, are driving anxiety. So anxiety isn't about uncertainty, it's about the brain taking low probability threats seriously, sometimes too seriously. And this actually wasn't Plato. This was Masuda Okan, who's leading this project. Shout out to Masuda. And I think this general idea is fitting into my sort of larger scheme of things. And I think we can use these kinds of models and this kind of rigorous formal description of what it is that we're looking at to drive our search for genes, cell types, modulators, brain circuits, and even real-world behavior. So that's what my lab's doing.
Thank you, Drew. That was awesome. Or and you didn't even need the chicken. All right, so now I'd like to welcome Simon Goldberg, who was a graduate student, research assistant between 2010 and 2016, and is now a faculty member at SCHM since 2017. So welcome. Good.
Thank you, Regina. Well, it's so fun to see the wide range of interests that align with our little Venn diagram with Richie in the middle, and it's such a treat, and you get such a sense of, really, I think, the breadth of your interest, Richie, and your passion for science writ large, and variability in how fast his students can talk, too. So this will be the half-speed version. But I'll share a little bit about our work promoting well-being at scale, and it's really inspired by the imbalance that you see pictured here. On the one hand, we have the enormous public health burden associated with mental illness. We know that 20% of the US population suffers from a mental illness at any time. And on the other hand, we know that there's no universe where we'll have enough human providers to meet this need, not to mention the unequal access to those providers across the country and certainly across the world. And we have the conviction that digital technology may play an important role in righting this balance. And we had a morning mind-melting session led by these folks on AI, and this is not the AI version. So check back in 2 years where this is all AI superpowered. It's, I think, as Richie might say, an incontrovertible fact that digital technology is going to be part of the future of mental health care. So this is our little foray into that. So 2 questions that we're interested in is whether technology can expand access to evidence-based strategies. We've been focusing on the contemplative tradition, and whether there are innovations in the technology that can go beyond what's been historically possible with the human-delivered versions of these. So I just want to share a little bit from a study that we ran. I am thrilled that Heather is in the room. Heather Abercrombie was a main instigator of this project, and so grateful to you, Heather, for making it happen. We tested 2 different versions of the Healthy Minds program in this study, one that had meditation practice and psychoeducation, and one that had psychoeducation alone to a usual care condition. This is a classic dismantling design, and we were really putting our money out there on the meditation practice making a difference. And inspired by Richie, we had all sorts of wacky measures in this study, behavioral measures, biological measures, all sorts of self-report measures, video-based measures. Raz Picard's in the room and was a huge part of that aspect as well. And we did find evidence, for all you meditators in the room, you're not wasting your time with your meditation. We did find evidence that meditation practice produced additional benefits beyond the psychoeducation alone. Although, as you can see in the figure, the psychoeducation alone, or what I think the Tibetan Buddhists would call the view, also was therapeutic relative to a usual care condition. And we see this across many of the different measure types that we assess. But we do see differentiation at a 3-month follow-up. And this was most pronounced for those who were more depressed to begin with. So it really seems like it makes a difference in some groups. We also see that there were changes in spontaneous language that people were using. So negative affect words and first-person pronouns are two aspects of language that we know have been linked to depression. And we see evidence that this meditation-based intervention does produce changes in that objective marker. But one of the things I really wanted to highlight that I think Richie has given to so many of us is the gift of fearlessness. So this is my punchline. These are, there you go, for all the neuroscientists out there. And in our study, this happened when we promised our funders that we would collect about 3,000 giant kits of stool samples. And we had no idea where we were going to put these 3,000 giant kits of stool samples. But Richie said, as he said to many of us, we'll figure it out. And indeed, as often happens, that's how I felt when he told me that. Somehow, it often does work out. I won't say it always works out, but it often does work out. And thanks to collaborators actually here at WID, Joe Handelsman and Margaret Thairu and others, we were able to have a much smaller kit, and we were able to see changes induced by the meditation intervention and, you might notice, the psychoeducation intervention on microbiome, in particular in pathways associated with short-chain fatty acid biosynthesis. So we think this is super cool, and as to our knowledge, the first data showing this from a digital intervention. So thank you, Richie. Thank you to the village who made this work possible, and thank you for organizing this session.
Fantastic. Thank you, Simon. So now I'd like to welcome Dan Grupe, who was a postdoc between 2013 and 2016, a staff scientist from 16 to 2023, and is now a research assistant professor in the center. So welcome, Dan.
So as I was thinking about what to share today, I was at the same time preparing a lecture for a class that we're teaching this summer at Oak Hill Correctional, which is a men's minimum security prison 20 minutes south of campus. And the lecture for that day was on gratitude, and that felt like a sign and a signal that should be what I talk about today. Gratitude, Robert Emmons has described as stemming from this perception of a personal positive outcome that's not necessarily of our own doing, not something that we can take credit for due to the actions of another person. And I think this is an audience that appreciates the empirically demonstrated benefits of gratitude, both giving and receiving gratitude. So it just felt like an appropriate topic to kind of orient this short talk around. So my work with Richie began about 15 years ago. My advisor, Jack, and I were working with Richie and Joe Fielgash, who's in the room, and others on a meditation study with trauma-exposed war veterans from Iraq and Afghanistan. And we were able to do this intervention because we had this beautiful meditation room over at the Weissman Center. And I'm just so deeply grateful that when we moved to our new home on West Wash, that Richie and Isa and others who were involved in that design insisted, again, on having a meditation space in the building, because this room has really become the heart of the work that we're doing now. This growing partnership that we have with people who have experienced incarceration, which I see as this through line that links this early work with veterans all the way up to this work that we're doing now. The other person who connects all of that work together, from early work with veterans to work that we subsequently did with police officers and meditation training, and our work with justice-impacted people, is Carmen Alonso, who many of you know, really skilled meditation teacher who her experiences teaching meditation in prisons and sharing those back with me and others was really one of the key inspirational factors for me in completely shifting the focus of my career to think about how can we promote more flourishing for people who have been impacted by incarceration. So back in 2023, Carmen and I started working with Christy Wilson Mendenhall to build out an 8-week meditation-based intervention. It's an in-person intervention with some digital support for people who are coming home back to the community. Thanks to our amazing community partners, including especially Deb Mahar, Aaron Hicks. We're in the middle of an NIH-funded clinical trial, small pilot clinical trial. We've run 4 cohorts now through this 8-week model, and this study is being managed by the really fantastic Mia Lauder, who's also pictured here. So 2 early data points, just a little snippet of data. You're just going to get a taste. That really reveal how much place, this particular place, but also the community that's supported through place matters for healing. So we developed a 12-item measure of safety, dignity, and belonging, which the author, Prentice Hemphill, has really highlighted as these are key ingredients, these key contextual factors that need to be in place for deep healing to occur. And participants in our first 2 groups where we administered this measure consistently report these extremely high, often or always feeling these experiences of safety, experiences of belonging, experiences of feeling dignified in this space. And those effects are increasing as the group goes on, as you might expect. And then just a qualitative data point from one of our focus groups and a participant naming this space and this group in this context as being a safe and almost sacred space in which to practice. And that word sacred, I just wanted to highlight because you don't always see this in the clinical trials literature. It might seem incompatible with this kind of work, but this work is different. So for people who don't always experience safety and dignity and belonging in their everyday life, it's not surprising that when these groups end, people want to keep on going. So in direct response to community requests, we started in late 2024 holding these drop-in sessions. We hold a dinner, we invite people to come by for an hour of meditation practice. We usually have about 15 to 20 people who show up. Some people have been out in the community for 20 years. Other people, we've had multiple occasions. People come home on Tuesday. It's the day that people get released from prison in Wisconsin. Wednesday night, they're sitting in a circle together practicing. This spring, we just ran our first weekend healing-focused retreat for our community partners out of Holy Wisdom, and we also developed and just finished the first iteration of a Train the Trainer program, where again, in direct response to people who complete this program and say, I want to share this with people who I know and who I care about and who I love because it's been impactful for me. So we're empowering people to be able to bring these practices to their peers, both in the community and in prison. So that brings us back to this project, our newest project at Oak Hill Correctional, working with this amazing grad student. I got the chicken. This was my goal, is to get the chicken.
We're grateful.
Patrick Nolan. We're working together to co-design a version of the Art and Science of Human Flourishing, which is taught here on the UW-Madison campus for incarcerated students. And we're working with 5 currently incarcerated students on this co-design process. We'll be teaching the class next spring. So I'm grateful in closing to the whole student flourishing team for designing this course around the idea that flourishing is something that we all have the capacity for, regardless of our life circumstances or where we're currently at. Also grateful to Richie for creating this whole center that's based on the premise that human flourishing is something that's worthy of scientific inquiry, and for inspiring me and so many others to take this up as our life's work. So in your words, Richie, a deep bow of gratitude for a career of really inspirational work and many years to come. Thank you.
It is so delightful to be here introducing all of these wonderful people, many of whom are friends, colleagues, and I am delighted to, I think for the first time, that I get to introduce Dr. Aaron Heller.
Okay, it's great to be here. Great to see you, Richie, and Susan, and Nisa, and everyone. So I'm going to start by trying to sprinkle in a few Richie-isms and notes that I took from my time as a graduate student here. And first is the importance of integrating research across species. the Health Emotion Symposium was a huge part of my development, my intellectual development, and understanding and appreciating how important it is to study and understand what's happening at the rodent level, and applying that thinking and data at the human level is critical. And so one of the key features of that that's influenced my work in my lab at the University of Miami is the importance of enriched environments. So you see here on the left, rodents housed in what are called typical cages, which basically don't have anything fun in them. And on the right, you have what are called enriched environments, which are much more akin to what's actually out in the wild. Not that there's hamster wheels out in the wild, but much more diverse stimuli, opportunities for exploration. And that's the key part of what an enriched environment is the opportunity for exploration, having diverse experiences and being able to grow. And one way to measure that is kind of developed by this paper that came out about 12, 13 years ago by Ullman Lindenberger and Gerd Kemperman, is using a metric from physics, roaming entropy, to study the degree of exploration or the degree of experiential diversity. That in this case, these genetically identical rodents affixed with RFID transponders on their hind were able to explore this enriched cage for about a month. And at the end of it, even though these rodents were genetically identical, they observed individual differences. And individual differences are a crucible in theory construction, as written by Benton Underwood in 1975.
What journal was it from?
American Psychologist. Well, yeah, I'm not sure of the volume or the issue. In any event, some of those rodents displayed high roaming entropy. That is, they distributed their time across space much more evenly. And other rodents, genetically identical, were much more sequestered in a corner of space. They didn't explore. They didn't experience as much experiential diversity across the month or so of the sampling period. And there were neurobiological differences between these rodents. Those rodents that were high in exploration, high in experiential diversity, were those that displayed higher levels of hippocampal neurogenesis in the dentate gyrus, which is important for well-being, for stress resilience, and depression. So along with a colleague, when I got to Miami, we deployed this in humans, right? We did GPS tracking in people, both in New York City and in Miami, and we found first that experiential diversity within person varies day to day. Some days we have high experiential diversity, and some days we have low. And in this project, we also asked them how happy were they. We asked them using experience sampling, how happy were they every other day over that 4-month period. And we found that days of high experiential diversity were associated with increases in positive affect. And that was quite specific. Their effects were kind of there for negative affect, but not as strong. We've replicated this in adolescence, so this is not an age effect. This is something that seems to be true across development, and this was even true in the early stages of the pandemic. For most people, even when the coronavirus was unknown of what the prognosis was or how it was transmitted, there were still positive effects between days of greater experiential diversity and increases in positive emotion. We have now partnered with the digital mental health company Talkspace to track people and across the United States. This is a pilot study. We now have about double this, across, recruiting people from 49 of the 50 states, having them do GPS tracking and experience sampling of emotion throughout seeking treatment for depression and anxiety. And we replicate our findings such that days of higher experiential diversity are associated with higher positive affect. Positive affect prospectively predicts changes in depression symptoms. Changes in experiential diversity also prospectively predict changes in depression symptoms. And if you think about a causal model about how this might occur, how well-being might be increased as a result of exploration, that these momentary experiences of boosts of positive emotion as a result of exploring, as a result of experiential diversity, might accumulate over time and decrease depression symptoms across weeks and months. So my real-life experiential diversity, just to conclude, I was born and raised in the Bay Area. I came to Wisconsin not knowing anything about Madison, and coming here has improved and increased my well-being. So whether that's taking photos out on the Weissman lawn with Regina, whether that's by chance wearing the same outfits as Javaris, whether that's taking a picture of Joe at one random night, or going to Fogo de Chao with Drew and Jamie. So thank you, Richie, and thank you, everyone.
This is absolutely wonderful to get to experience this experiential diversity as we go through the years. I am delighted to introduce our next speaker, who may look familiar to you as one of the other MCs. Regina Lapate was an undergraduate research assistant in 2004, staff from 2005 to 2008, graduate student from 2008 to 2015, and postdoc from 2015 to 2016. So we are delighted to welcome her here.
Thank you, Drew, and thank you all for being here and for having this wonderful event. So of the many enduring lessons and to the field and really that have been influential in my own research that Richie has taught us is underscoring that emotions are dynamic and that traditional divisions between subcortical emotion systems and cortical prefrontal cognitive systems are artificial and deeply fraught. And to many of his trainees, Richie has taught another lesson, which is to not let the methods that you're comfortable with constrain the research questions that you can ask. And so for my dissertation, Richie encouraged me to go into Brad Postel's lab to learn transcranial magnetic stimulation, or TMS, to try to establish causal inference in the processes that we were studying and the circuitry that we were studying for affect regulation and awareness. And what I didn't know at the time is that this new venture would give rise to what's now nearly half of my lab's ongoing research program in trying to understand the organization of prefrontal cortex in emotional processing and regulation. And so today I'll share an example of the types of research questions that we're pursuing. So we know that lateral prefrontal function is essential for temporally organized goal-directed behavior. And goal-directed behavior should incorporate emotional information into it, and ideally not just momentary signals, but emotional information that's integrated over time. So how is that process happening? So we know from work done in the organization of cognitive control in lateral PFC that the anteriormost region of lateral prefrontal cortex, the frontal polar cortex, supports temporally extended control signals. And this region also has privileged access to amygdala projections. And in some of my prior work, we've shown that this region holds emotional valence signals or representations during cognitive control tasks when that information is goal relevant. So we thought that putting these pieces of data together, the frontal polar cortex is perhaps ideally situated to integrate or at least track temporally integrated emotional information to guide goal-directed behavior and inform context-appropriate action, and that's what we tested in this experiment that I'll be sharing today. So to test this idea, we designed a new task that we call the duration different estimation task, or the DUDE task. That was led by my first and now senior PhD student, Mengzi Li. So in this task, participants were shown 4 emotional images, 2 positive and 2 negative, for a total of 12 seconds. And we varied the duration of the individual images such that half of the sequences were predominantly positive in valence and half of them predominantly negative. And that's what the participants were asked to tell us. And they did so according to a contextual action cue that they saw later in the trial that told them the mapping between the predominant valence that they held in mind and an action, a left or right button press. Participants can do this task well, and they benefit from the additional temporal evidence in favor of a valence that we provided them with in half the trials. Okay, so we're going to examine this canonical regions along the rostral-caudal axis of lateral PFC using a classifier. And what we find is that during the emotional sequences epoch, the frontal polar cortex tracks the predominant emotional valence, which is not the case for our other ROIs. And moreover, trial-wise, the strength of this classifier in frontal polar cortex tracks with or reflects the accumulated emotional valence evidence at the end of the sequence, which is again not the case for other frontal regions. Now, during the contextual action processing queue, when they can translate this emotional valence information into an action according to a rule, we see a shift along the rostrocaudal axis in this representational profile, where the rule is more robustly, this mapping information is more robustly decodable from midlateral prefrontal cortex, and the action that they can now begin to prepare is most robustly decodable from premotor cortex, as you'd expect, suggesting again a shift along this rostrocaudal axis in the flow of information from integrated emotional evidence into an action. But of course, this alone doesn't establish causal inference regarding frontal polar participation in this process. So what we did is we brought the same participants back for 3 TMS sessions, followed immediately by an fMRI scanning of the same task. And we targeted their brains and frontal polar cortex, midlateral PFC, and a control region using our previously validated approach that we called information-based TMS, where we're going to target the specific individualized location of the maximal decoding of that particular task property, in this case, emotional valence in frontal polar cortex. And what we find is that after targeting these regions with this inhibitory TMS protocol, participants are indeed significantly impaired in performing this task after frontal polar stimulation compared to our other sites. So I think the summary, you probably still have the conclusions in your working memory in mind, but in essence, frontal polar cortex seems to indeed integrate or at least track this temporally extended emotional evidence over time to guide action, and I think this importantly extends theories of the organization of cognitive control in prefrontal cortex to incorporate emotional signals. So thank you, Richie, and thank you all.
I feel so grateful to be able to introduce all of these people, many of whom I have known for years and years and years, and the next speaker is no exception, though I don't know how these links are going to load. Uh-oh. Yeah, there you go. You're going to have to drag it. Jeff Maxwell, otherwise known as Max, graduate student in the lab from 2000 to 2006, and postdoc from 2006 to 2008. My one-time office mate, and probably the least associated with academia here.
So before I arrived at Richie's lab, I did a lot of work with schools out in Northern California and the Vacaville Police Department with kids who were getting in trouble for truancy, who were at risk of expulsion, helping to basically find alternative paths. So probably unsurprisingly to Richie, it was maybe half the way into grad school, I was like, hey, I want to do this NSF program instead of being paid on an R01 grant or doing the typical research or teaching assistantship, where it was building STEM curricula for inner city schools in Milwaukee. Schools that were underperforming. And we had to develop whole different approaches to this. And in true Richie fashion, he asked a couple of questions, made sure that I was counting the extra work it was going to take, probably with a little bit of an impact on grad school, and then was fully supportive of the vision. And so for those who I was in school with, I don't know if you remember, but I spent a lot of time actually doing that 1 year instead of grad school. Well, since that time, my wife and I have done work in Haiti and in Zambia and in Kenya and quite a bit throughout the DC area. I think we've got 6 students now who have half or full of their college tuition being paid for by companies because their family couldn't afford it, but this way they don't have to take out loans in exchange for continuing service agreements, which means they have guaranteed jobs when they get out, which is also pretty cool. Anyway, that's all coalesced into what I wanted to share today, because we are in the process of going live. Let's see, where do I drag it? Sorry. Over there. Okay, there we go. Cool. Now, can I click? So the idea Amy and I had, like, while we're doing all this, we're like, man, this really, there's no town square. There's no central place where students can find anything. Every set of parents is using Google to try and find service hours. Every school counselor is trying to arrange things with local nonprofits, internships. There were a few government ones, but we didn't even know if other ones existed. But when we started talking to companies and eventually searching their sites, like buried 3 layers deep on LinkedIn or on company sites, there are internships out there. All the information was there, but scattered. And so I was really glad at that moment that Richie had convinced me to go the non-clinical route and spend all that time with messy, unstructured data. Because getting a handle on this, part of it was finding it, but then getting a handle on this and organizing it and bringing it all together was a pretty monumental task. And that's what I've been spending pretty much nights and weekends for the past couple of years doing. But now with Varsity Force, it's all in one spot. This is live, and we already just got 3 companies. We're not charging anyone anything for this, like nothing. We're doing it out of pocket, just as something to give to the students. Maybe eventually some companies, if we're in the schools, will want to partner with us and kind of donate as sponsors or kind of things like that. But this is really just a give back to the community. And I never use the word flourish, but we got the website opportunities&hope.com, jobsnotdebt.com. We've been acquiring some of this over time. Some of them we've had to negotiate and get, but the way we think of it is opportunities and hope. But when you're talking about flourishing, it's the same end goal. We think that by connecting families and schools and students with these kind of opportunities, it can get them thinking about next year and 2 years beyond, and then downstream. Some of what we have is the ability to explore college majors of all kinds, talk about, can you get employment in that exact field, or would you need to go on to grad school just so it's eyes wide open? Similar things to that for, well, let's see. I'm not going to try and click through this anymore, so you get the idea. But yeah, so anyway, if you guys know anyone who's in DC, Maryland, or Virginia, no strings attached. This is live. It's running. We're going to keep it up. Going to incorporate it as a 501c3. And if possible, in maybe 2 years, when we make a little headway on our own kids who are going into college, Amy's going to try and do this full time to switch to it. But it's all just from an opportunity of trying to help people flourish. Yeah, and thank you for that.
We get a break. All right, we're going to be back here at 2:16 promptly with our next speaker. I believe it says break with refreshments. So I expect everybody to be here promptly and refreshed. I'm looking at you, Carien. All right. Thank you all to our speakers.
I really enjoyed your talk. You didn't comment on it explicitly, but it really connected. Announcement number one is that due to the unfortunate air particulate quality, the Saturday picnic has been moved from Violas to CHM. Second quick announcement is that we're going to, once we clear the room, I would like everyone who's presented here today at the Blitz to gather with Richie, and we're going to take one group photograph so we can remember that aspect of this sort of Festschrift. And with that, I'm going to hand it over to Regina.
All right, welcome back, everyone. So now it's my pleasure to call to the stage Matt Hirshberg, who was a graduate student from 2000 in the center 2015 to 17, a postdoc from 2017 to 2021, and is now a scientist and research professor at the center. So welcome, Matt.
So one time, I don't remember how we got into this conversation, but somehow Richie and I were talking, and he told me about, he was at a conference and had some very prestigious talk to give early in his career, and he had worked up, you know, a huge speech, so to speak, like a written text, and I think said he was quite nervous, and a senior mentor was next to him and looked at him and was sort of like, put that down, you've got this. So Richie put it down, and I don't think he's ever picked up a script since then. So I'm gonna try to channel Richie here. We'll see how it goes. So I thought as a frame, I'd talk about what works for whom and when. I mostly do intervention research, and I think if we look at the arc of contemplative science research, a lot of what we've done is what works. And in particular, like what interventions work. We've also paid a bit of attention to the other side of which is what are the outcomes that we think these things affect. So there's been a lot of clinical science, depression, anxiety, et cetera, but also some other outcomes about social relationships and that sort of thing. But predominantly, that's the sort of, I think that is the majority of the research that's been done. I think we've asked less, although people are starting to, about how can these sorts of trainings affect or address sort of real-world emergent problems that people are facing now. So this is like really about application, right? Like how can training the mind in the ways that we've studied address some of the unique challenges of today? So this was a theoretical paper that we wrote with Richie, making the argument that actually well-being skills, the sorts of skills that we know are trained in the types of interventions that we've been studying, affect characteristics that actually, particularly if they're targeted towards a domain like digital hygiene, may actually really be able to support kids in using digital technology in ways that are adaptive and less maladaptive. Because of course, digital technology is here, it's not going anywhere. So real world applications, I think, is one of the exciting areas where we're moving forward. So for whom? This is a question that we've addressed on one level. So when we run intervention studies, we'll often look for moderation effects. What this means is the effectiveness of an intervention different, say, based on gender identity or racial identity or age? So that type of analysis we do we do that frequently, and we often find either there's no moderation effect, which is kind of a good thing. It's sort of like these work in general for most people. And where we find a moderation effect, the most consistent one is we often see the best benefits, the largest benefits for people who are suffering the most. So those with the highest levels of anxiety, depression seem to benefit the most. This is a more contemporary approach. We did a heterogeneity analysis. This was led by one of Simon's graduate students. The data are the study that Leandro presented at the beginning with Mexican healthcare providers. So we asked, are there differences in the effectiveness of the intervention for people? The answer to that is yes. And then we asked, can we predict who's likely to benefit based on all the variables that we collect at baseline? And what's really cool about this analysis is that it turns out with just 20 items. So we measured 185 things, I think, at baseline. And 20 items do better than all 185 items combined in predicting who's going to benefit. And what you can see is almost everybody benefits. 80% of people benefit a small magnitude or more, but 20% don't benefit at all. So we probably don't want to give this intervention to those people. These people we definitely want to give this intervention to, and we seem to be able to predict that with quite good accuracy just based on about 2 minutes of self-report items. So this is exciting because in the future, this is sort of offers the potential for precision medicine. And then for whom and when, these are questions we also haven't paid that much attention to. These data Richie spoke to last night. These are data from teachers and students in Louisville, Kentucky. And each circle over here is the average learning gains of a classroom. So there's 15 to 25 students in each of these bubbles. Each bubble is a teacher. And what we can see is that the orange here are the teachers that were assigned to the Healthy Minds program, this digital app we used. And on average, their students made statistically significant gains in end-of-year math learning. This is what Richie presented. Just a little context, the scientists in this room might be looking at this critically and saying, like, these are super tiny effects. How is this important? This is the average, the last 30 years of educational research, federally funded, rigorous research, trying to achieve improvements on these outcomes. There's no effect, basically. There's an average effect that is very small. These are the effects when the teacher gets 4 weeks of intervention, so no intervention for the student. So there's some transmission here that's going on. So for whom and when is an interesting question, because for whom might be for the teacher and not for the students when they're students, and actually by intervening with the teacher, we may be able to benefit students. So we often think about for whom and when and what as separate things, but actually I think these are interacting in really interesting ways. Do I have any time left? No. Okay, thank you.
Thank you, Matt. This was great. All right, so now it is my pleasure to introduce Tammi Kral, who was a graduate student, or started as a staff from 2010 to 2013, then a graduate student from 2013 to 2020, a staff, and now is a staff scientist and director of science at Human from 2021 until the present day. So welcome, Tammi.
Hi, everyone. It is just really thrilling to be here. So I want to thank again the organizers. It's been so wonderful to hear all the presentations and see so many friendly faces. My career with Richie and really otherwise started in a lab, specifically at the Weizmann Lab for Brain Imaging and Behavior, where at the time, just before I met Richie, I was working in an affective neuroscience lab studying brain mechanisms underlying symptoms of anxiety and depression. And it was during that time when I heard about this opportunity for a post-bac staff job working on one of the first randomized controlled trials of MBSR, looking at how meditation impacts the brain. And I was like, this is amazing. As an undergrad, I was really interested in hearing Richie's first results of brain differences in expert meditators and studying the biology of consciousness and all those sorts of cool things. And so when the opportunity arose, I jumped at it. And really, that was the beginning of what's now 15 years as of this year working with Richie on the science of well-being and flourishing. And so after joining that study as a post-bac member, I spent a number of years at the Center of Healthy Minds doing my graduate work in affective and contemplative neuroscience. And for the past 5 years, I've been bringing well-being science out of the lab and into the world with my work at the nonprofit Human. And so today I'm going to talk a little bit about that work that we've been doing, just a little sprinkling of various things. There are a lot of cool collaborations we've been doing with the Center of Healthy Minds that I won't get to talk about today, but would be happy and love the opportunity to talk to you more about that later today. So one of the ways that we're bringing well-being science out to the world is through partnerships with nonprofits like the Wellbeing Project and the Center for Healthy Minds, where we're conducting a worldwide survey of change makers across many different sectors to really try to establish a global baseline of how are change makers doing, what are the factors associated with resilience, with ill-being, and with well-being. And as you can see in the dark, kind of bluish purple, those are where we have the most responses. We have over 10,000 responses in total. We just recently closed the survey. And so the dark regions have at least 1,000 responses, and then the lightest regions have a couple dozen, with many falling somewhere between that. So we have lots of really great data, and for this survey, we wanted to use well-validated measures that were available in most, if not all, of the 13 languages. And we had a couple where we got professional translations. And so those included the WHO 5 Wellbeing Index, as well as established measures of anxiety and depression symptoms, burnout, and general stress. And so I'm not going to present any actual results today. So this is truly a teaser, but please come find me. We just started taking an initial peek at the data, and I would love to tell you a little bit about what we're starting to see. Another area of our work is about bringing well-being science to industry. And so one of the first sectors where we have been doing this work is in hospitality. So we just recently published a white paper that we did in collaboration with Hyatt Miraval, where we did one of the first really actually rigorous longitudinal studies of the impact of a wellness retreat on well-being and stress. And so this was a longitudinal study that was naturalistic. And so this was real guests who were booking a stay were invited to participate in a study where the only eligibility criteria were booking a stay with at least 14 days in advance so we could conduct a baseline survey. And what really differentiates this from your standard industry customer satisfaction survey is this longitudinal design and the incorporation of established validated measures like the perceived stress scale. And so participants would complete a survey at least it was sent out 14 days prior to their stay. They had to complete the baseline survey before they arrived. And then 14 days after their departure, they were sent a follow-up survey. And then again, 60 days following departure. And what was really exciting is that we found significant effects across multiple well-being measures, mindfulness skills, and reduced stress that were significant 14 days following their stay and remain significant 60 days following their stay. So this is really cool compared to your kind of standard industry customer satisfaction survey that's sent out like the day you depart, right? Like, how are you feeling? So, well, you're probably feeling good because you just finished your stay. So really exciting to see these lasting effects and to be able to bring both this rigorous science into industry and also a focus on well-being and flourishing. The other area of work I'd like to highlight is our well-being measures development. And I'm happy to say that self-report is no longer taboo. So we have been developing a number of self-report measures of well-being. In addition to that, we have a suite of these gamified mobile measures that are based on either self-report or behavioral measures from the psychological literature that we have gamified and conducted a series of studies to start scientifically validating them as measurement tools. And so the first of those studies was recently published in collaboration with scientists at the Center for Healthy Minds, and that is establishing initial validity of single-item self-report measures of well-being that can be used in experience sampling studies. So with that, I was waiting for the chicken. I'd like to thank you, my colleagues at Human, collaborators at the Center for Healthy Minds. Special shout out to Raquel Tatar for making everything possible that we do scientifically at Human, and a deep bow of gratitude to Richie for many years of wonderful mentorship and hopefully many more years to come.
Delightful. It is my pleasure to introduce our next speaker, Robin Nusslock, who is an undergraduate research assistant from 1997 to 1999, and then a graduate student from 2003 to 2009. So let's do this, Robin. Hey.
Wow, what a privilege to be here. This is a picture of me when I was in my mid-20s. It was actually about a week before I met Richie in the Laboratory for Affective Neuroscience, as it was presently named at the time. And they say that change is possible. You all say that change is possible. I would suggest that change is possible, although in the case of my appearance, I might like my older self. At the time of this photograph, I was majoring in Buddhism and philosophy, and was and still am a seeker of the mind, but I was a bit of a wandering sadhu at the time. I wasn't exactly sure how I wanted to study the mind. And then I saw this advertisement, One Special Day for Me, let's advance the slides, for a job or research assistantship scrubbing electrodes for the MBSR study with John Kabat-Zinn. And when I walked into this lab in the first week, I knew precisely that this is where I wanted to be and this is what I wanted to do for the rest of my life. And so I think of all of the people that were there, Diego, Heather, Abercrombie, Alex, Chris Larson, and Richie. I mean, there's really no group of people that have had a more positive effect on my life than you all, and I just want to take this opportunity to so publicly thank you for giving me that direction. And Richie, thank you. You continue to be an inspiration for me in all things that I do. I'm a professor at Northwestern University where we use EEG and MRI and other technologies to study how the brain creates emotion and how those brain systems are implicated in both mental and physical health. And in the last period of time, I've become particularly interested in this concept of whole person health. That is, how is it that the brain and the body communicate with each other to generate both risk and resiliency for mental and physical health problems? This is work that I'm doing with Greg Miller and Edith Chen at Northwestern University. In particular, we're interested in these statistics for why is it that somebody who makes it to the age of 40 and grew up in adversity, why do they die on average 15 years younger than somebody who didn't grow up under conditions of adversity? What are the brain-body pathways that really drive these important mental and physical health outcomes? So we particularly focus on signaling between the brain and the immune system. But more recently, we're also looking at signaling between the brain and the heart, taking kind of a multi-organ, multi-systems perspective. So one of the themes of my work really is multi-systems perspectives, where we're interested in looking not only at multiple organ systems, but also looking at the individual, the family, and the societal implications of and influences of different levels of analysis on the brain and body. One of the things that I've really learned from you all is that well-being is a skill, and that internal and external factors not only can affect whether somebody's at risk for mental health problems, but also their resiliency to mental health problems. So I just want to present one study. This is one of several studies, many studies that I've run with my colleague Greg Miller. And really looking at, in this case, internal factors that are predictors of a resiliency to mental and physical health outcomes. So this is a study that we're in the longitudinal phases of it. This is a map of the city of Chicago, and these are all of the murders that happened in the city of Chicago for the 5 years prior to this particular MRI scan. And so we're able to really get a sense of the environmental adversity with which somebody actually grew up. And so we took measures of central executive network functional connectivity, which we know is implicated in emotion regulation, prior to also drawing blood. And I just want to point this one figure out so that on the x-axis or the y-axis, what we have is cytokine release, so that's a composite of inflammation, with high values here reflecting more cytokine release and low values here reflecting less cytokine release. And chronic inflammation we know is implicated in both mental and physical health outcomes. And then we divide, for just kind of presentation purposes, individuals into low versus middle versus high network connectivity in the central executive network. And what we find is that, as expected, individuals with low network connectivity and high exposure to neighborhood adversity show heightened inflammation. By contrast, individuals with high network connectivity are entirely protected from the consequences of exposure to early life adversity. And so we're very interested in digging into this in more detail, and we're looking at environmental and psychological and other biological factors that may help protect people from adversity, and that's a major theme of my research program. The last thing I want to highlight, this is actually a picture of Richie on his first trip to India, where I think Roshi Joan might have been there, and maybe Cliff, taking some of the equipment to India and actually starting to study the monastics in a real environment. And one of the great privileges of my life at present is, in my own way, to kind of carry on that tradition, and we're actually, instead of studying the monastics, we're actually trying to to train the monastics to do research themselves. So we've now built several neuroscience laboratories in India and training monastics over there. Actually, Jampa Taksho is here, one of the monastics from Bhutan. And the idea is to actually start to center contemplative science in the monastic community as opposed to have it just be people who look like me who are doing these studies. And so, Richie, what you've done in this field of birthing contemplative science is inspiring, and I'm really excited for our future collaborations in this area. Thank you so much.
Thank you all for keeping on schedule. It is my delight to introduce our next speaker here, Carien van Reekum, who was a postdoctoral fellow from 2000 to 2002, a postdoctoral research associate from 2002 to 2003, and then an assistant/associate scientist from 2003 until 2007, and was one of the people who I consider to be one of the models for how to exist in Richie's lab.
Thank you all. Cheers. Listen, I'm going to briefly cover how my time in medicine really has influenced the research that I'm trying to do now, which moves towards emotion flexibility. Very early work, and I'm really both nervous and really curious about the input that you may have on some of the ideas that I present here. But let's just first start with a couple of Richie's wisdoms. There are many things that I've learned from Richie. But here are two of the numerous wisdoms that Richie bestowed on me that have really influenced my academic program so far. Study of individual differences is crucial to our understanding of basic and biological mechanisms. I really came to the lab thinking that the study of individual differences was really left for clinical psychologists, but Richie showed me that they really inform the basic biological and psychological mechanisms. I attributed it to Paul Meal. I'm wrong. Aaron Heller. What was it again? Underwood. Thank you very much. Okay. A direct quote. A study not worth doing is not worth doing well. I'll return to both of those aspects. Whilst in Richie's lab, I had the great opportunity to work with Heather Urry and Tom Johnston on the process of emotion regulation, or more specifically on reappraisal. So we took the individual differences approach to heart and really focused on some of the brain mechanisms that support that. So we now have a much better understanding using this individual differences approach on the brain mechanisms that support well-being, in this case in older adults, and how some of the recruitment of some of those systems can be counterproductive in individuals with major depressive disorders. We also took the individual differences approach to understand some of the more mechanistic, methodological issues surrounding some of the brain clusters that we observed here in this case, whether they reflect cognitive demand or cognitive effort, led by Heather, or attentional control reflected in gaze fixations. That really brought me to this quite recent paper that I work on with my colleagues at the moment on the revision, where we applied fMRI constrained principal component analysis to really understand the different components of cognitive modes that support reappraisal. Two of them here are highlighted: a multiple demand mode that is particularly engaged when people are increasing their emotion using reappraisal, and a reevaluation network really reflecting specifically, or among other, the decreasing. So the goal of regulation really, really matters. That brings me to one of the topics that I want to pick up again, and that's aging. Flexibility as we age. We know that when we get older, we experience brain atrophy, cognitive functioning goes down, but emotion function remains relatively intact. That it happens through compensation or neuroplasticity, if you want. Older adults use different neural pathways to achieve the same effect and maintain emotional function despite perhaps some memory issues that they may be experiencing. That is also associated with using less mentally taxing emotion regulation strategies whilst maintaining all that. I'm going to not talk about the tipping point because I only have 2 minutes left. So the grant that I really want to work on later this year is really focusing on emotion flexibility in the aging brain. What are the compensatory mechanisms? And in addition to just taking classic fMRI data, really want to get back into a bit of the neurochemistry. This model by Mara Mather, it's the autonomic compensation model that some of you may be familiar with. She really focuses on the extent to which aging is associated with locus coeruleus integrity loss, affecting noradrenergic systems, and is associated with higher arousal, higher sympathetic arousal in older adults. She maintains, as part of this model, that the recruitment of rostral ACC areas, among other, are really used as compensation to trigger parasympathetic activity in order to counteract that noradrenergic activity. So that's a system that I'd like to focus on in some of the work. But in addition, really, my smart colleagues Tiffany Bell and Anastasia Christakou are really experts in MRS and have been shown how important choline is also in human flexibility. And we know how choline is also, acetylcholine is important, for instance, in Alzheimer's. So the grant that I want to be working on, and I would love your input on that, is really sort of pitching those two systems against one another using both longitudinal existing data and collecting some new data and really examining the extent to which that underlies emotion flexibility in some of the compensatory processes. So I'll leave it at that. And I just want to thank you. I am the scientist I am today, largely thanks to Richie, who has provided me with the opportunity to be in the lab and to work with the fantastic colleagues and a lot of you whom I'm so pleased to call friends. I've learned so much from you. Thank you, thank you, thank you.
Thank you, Carien. That was wonderful. So now it is my pleasure to welcome here Melissa Rosenkranz, who was a staff from 1997 to 2001, a PhD student from 2001 to 2008, a postdoc from 2008 to 2010, a scientist from 2010 to 2019, an assistant professor from 19 to 2025, and now is an associate professor at the center from starting 2025. Welcome, Melissa.
Do I get a prize or something for that? I should.
No.
I have a chance later to give some personal remarks to Richie, so I'm going to stick to science now. And I am gunning for 2 squawks because I'm going to cover 25 years of science with Richie in 5 minutes. My Richie-ism that I have really taken to heart through the whole of my career is, actually, I don't even know if you said this or if this is somebody else's quote, but in nature, hybrid species are sterile, but Francis Crick, thank you very much. See, I did not inherit this ability to remember who said what, when, and where. I just always stand next to Richie so he can tell me. But to finish that quote, but in science, that is the most fertile ground. And so the overarching theme of my work is to understand the biology behind mind-brain-immune interactions. And specifically, I'm trying to understand how the contents of our mind impact the function of our bodies, and how do the activities of the immune system color the lens through which we experience the world. And we see these relationships manifest in the high concordance of chronic inflammation with mood and anxiety disorders. And in much of this work, I've got notes to keep myself on track here because otherwise I wouldn't. I've done much of this work using allergic asthma as a model. Asthma is a chronic inflammatory disease of the airways. It affects about 10% of the population. And it makes a really extraordinary model for this work because it's highly prevalent. It persists across the lifespan. And importantly, we can safely provoke symptoms of asthma in an experimental setting. And using this model, we've studied the impact of airway inflammation on emotion processing using brain imaging tools collected at before and after an allergen challenge. And we've been able to identify specific immune pathways that are activated by exposure to allergen in patients with asthma that shape the activity of specific neural circuits involved in determining our experience. So in other words, these changes shape what stimuli in our environments we pay attention to and how we interpret them. We've also done the inverse, where we activate stress and emotion-responsive brain networks using a laboratory stressor, and then measure the activation in those neural circuits using the tools of brain imaging. And then we measure how those changes in the brain impact the way that the lung responds to exposure to allergen. And what we found is that the same immune and neural pathways are involved in this brain to lung direction of communication that are involved in the lung to brain direction of communication, likely united by the actions of the sympathetic nervous system. So you may be wondering, since we've shown that stress and emotion can exacerbate inflammation in the lung, can reducing stress or effective emotion regulation reduce airway inflammation in the lung? And I wondered the same thing, and I tested this using a mindfulness-based stress reduction intervention in patients with asthma. And indeed, they can. So we were able to show that 8 weeks of MBSR training led to a sustained improvement in both asthma control as well as a reduction in airway inflammation. For Alex Shackman, the N on this is about 72 participants. He called me out on that just by looking at my slides. And at the same time as I was doing this work, I began to wonder what the long-term consequences of this relationship might be, because in the animal literature, trends were emerging showing that inflammation in the body could provoke inflammation in the brain. And indeed, together with a colleague, Christy Bartles in the Department of Medicine, we showed that the 2-year incidence of a new Alzheimer's disease diagnosis was 18 to 24% higher for patients with asthma compared to a non-inflammatory control group, and the prevalence was 48% higher. So I started to look for evidence of this in my own data. And what I found was a shockingly large and widespread difference in white matter microstructure between cognitively healthy, middle-aged adults with asthma compared to a matched non-asthma control group. These changes were correlated with the severity of asthma and processing speed. They were also correlated with the concentration of a protein measured in plasma that indexes the reactivity of an immune cell in the brain called astrocytes. And this suggests that at least a portion of these changes in white matter microstructure could be attributed to neuroinflammation, or were likely related to neuroinflammation at any rate. We also showed in a separate sample, yep, there goes my one squawk. We showed that these brain microstructural changes in both white matter as well as in gray matter of the frontal cortex were associated with a more rapid decline in cognitive function over time in patients with asthma. So right now, we're currently following up this work with a study that was specifically designed to determine if airway inflammation in asthma provokes neuroinflammation in a multimodal neuroimaging study with this amazing interdisciplinary team of collaborators that I wouldn't have known how to build or wrangle without the training that I got from my fearless mentor, Richie. Thank you so much. I didn't make it to 2 squawks. Thank you.
Fantastic. Thank you so much, Melissa. All right, so now this will take me a minute because I believe it's the longest set of introductory notes that I have. It's my pleasure to introduce Cliff Saron, who was an undergraduate student with Richie from 1974 to 1975, and then a full-time research assistant from 75 to 76, then from 1977 to 1985, research associate in Richie's lab in New York, State University of New York, purchased, then from 1985 to 1989, an instrumentation integrator at the University of Wisconsin here, from 1989 to 92, technical consultant, and long-term collaborator. Okay.
So I wouldn't be here, and I have no idea how to conceptualize my life without Richie. So Susan Davidson took this picture when I visited them in Maine on vacation in 1978, almost 50 years ago. But most of the time you could find Richie and I in this state-of-the-art, computerless, 8-channel EEG laboratory at the State University of New York at Purchase. But several years later, we upgraded it to 16 channels So you could see we were able to use electrocaps, and I chose this image to introduce Richie to the Dalai Lama when in 1990 he extraordinarily invited me to fill in for him in the Third Mind in Life conference, and I tongue-in-cheek indicated that this was a titled image, Hasidic Aliens. And the Dalai Lama looked at me like, what the fuck? And I said, Jews from outer space. And then everyone cracked up. And during that meeting with Alan Wallace and Francisco Varela, we resolved to do a longitude or a study in the foothills above Dharamsala that Robin just alluded to, a field research project on the effects of Tibetan mental training on monks, and we, of course, involved Richie immediately in envisioning this project. So here he is after an all-night train ride through the Punjab from Delhi, and to the right were porters pushing half of the 1,100 pounds of equipment we brought. We also went to see these monks going up goat trails in the hills of Baksu Mountain, and here is Richie and a monk's attendant, Greg Simpson and Alan Wallace, our translator, outside the hut of Geshe Topke, who gave us a discourse on the nature of compassion that changed the course of our research. And it was that afternoon that Richie committed to bringing the cause of compassion into the field of mainstream psychological science. Many monks have been coming through Wisconsin for many years, but the first monk was Geshe Donyo in 1993, brought by Jose Cabezon. And here he is, Richie is showing the lab, and believe it or not, IAPS pictures and Tibetan monks began in 1993. So my lab at UC Davis actually carries a thread of that afternoon with Geshe Topke. Fundamental research question has been how can we engage with and compassionately respond to our suffering and the suffering of the world as it comes to us, and kind of felt sense of common humanity inform answers to these questions. So a lot of the work that we're doing comes from the consequences of what we did in the Shamatha project that are summarized in this paper from a couple of years ago. And a current project relevant to this theme is the Pathways to Compassion project, looking at Zoom-administered compassion cultivation training and MBSR compared to a non-training control group in a longitudinal design. And the main task involves participants' responses to affective images of both valence and motivational direction, and the IAPS is no more. We've spent the last 6 years, and you can get out your phones or come see me, click on this QR code, creating a new high-resolution thematically organized stimulus set for affective neuroscience. It's 7 categories, thematically organized and diversity informed. Here are the categories for the suffering images, just to give you an example. And in a recently published norming study, you see on the left that there's uniform negative ratings for the negative categories. Harm is a victim and a perpetrator, suffering is a victim alone. But there's much more spread of the motivational direction. And we found that individuals higher in empathic concern rated more approach motivation in the responses to suffering and harm. And we've recently found that after compassion, but not mindfulness training, we see a very similar effect. So in the remaining overtime, I'm going to give you a taste of a passion project I've been involved with for the last 48 years, which is actually the last 3 years, repatriating digitally anthropological film that I was part of filming in 1978. And I'm going to leave you with a 50-second clip of Cook Island Maori dancers chanting a celebration I'll tell you about offline.
Thank you to Cliff. And it is my delight to introduce Tor Wager, who was a collaborator and actually, I think, never actually a member of the Davidson lab. So I think we can all learn something from him. Come on up.
I don't know how I made it up here, but thank you for adopting me as the honorary trainee. This is the title of my talk, Richie Fest. Cliff, I love those pictures. It's amazing. Back to text now. I wanted to talk for a minute about the Mind, Brain, Body, and Health Initiative. This is how I first met Richie. And this was a group that was pulled together by, it was a network of wonderful faculty and doctors, pulled together by Bob Rose, who is a stress biologist. And we had these heavyweights in cognitive and affective neuroscience, like Richie, Steve Coslin, my mentor, Ed Smith, John Cassiopo, and a whole cast of characters like David Spiegel, many others. And these were really serious scientists who were talking about these weird fringe topics like hypnosis. Can you study that? Meditation, all these kinds of things. And so I, as a young graduate student, thought, maybe I can actually study that. And it definitely changed the trajectory of my career, as you'll see, because it launched really the direction that I'm still taking today. And from this group, with a lot of great ideas and very small amounts of funding, came multiple research programs, including the asthma program that Melissa talked about and launched her and her career here. This work on loneliness and the first study by Steve Cole and John Cassiopo of gene expression changes with loneliness. And that launched a program that's still alive and well today and has flourished on human social genomics. Also, John Sheridan's work on stress biology. So does psychosocial stress, social disruption really influence the spleen and the immune system? And it does. We're following up on that in 2026 with brain imaging and transcriptomic studies, but I won't tell you about that today. And then there's placebo effects. So they published a wonderful paper with Jack Nishke on placebo effects in taste, in insular taste-related cortex, and our work on pain, showing that indeed, maybe just your expectations and beliefs about a treatment that you're going to receive impacts how your brain responds to and constructs the experience of pain. And this idea, I think what made this study important in part was it was a kind of answer to this question that has been, a big one in the literature and still is a question today. If you believe X something, does it really change your experience in an important and fundamental way? So studying the brain outcomes during pain experience was a way of doing that. A very short summary of some of the findings in the ensuing years. From one study, now we're up to meta-analyses of hundreds of people with individual participant analyses. So yes, if you believe something is going to help you, it reduces activity in many pain-related areas of the brain. It activates pain control systems in the brainstem, including the periaqueductal gray. This was a science paper from this last year. Apparently, you can activate even parts of the periaqueductal gray that correspond to particular body sites, which is amazing. It can change activity in the spinal cord in response to pain in both directions, up and down. It can cause endogenous opioid release and more. So we spent the next 20 years basically figuring out more about the landscape of whom does it work for whom, and why? And you can ask me more about that because there's a much more nuanced landscape. But a through line throughout all that is measuring the effects of interventions on brain systems. So that's an important thing, I think, for the field still. That led us, and this early placebo work led us to develop a research program where we're trying to build meaningful brain models of all kinds of affective experiences. So we're trying to develop models using machine learning and now AI tools, validate them, optimize them, and then we can test them on interventions, including placebo effects and psychosocial interventions, having a supportive, caring person, having your spouse hold your hand, mindfulness and meditation, and now even brain stimulation, which actually started from the Wisconsin Emotion Symposium a couple of years ago when I was chatting here with Alvaro Pasquale-Leone and was like, we can do temporal interference stimulation and start this. So now we have a whole line of research on that. It also supported these brain biomarkers related to pain, allow us to test mindfulness and meditation. So this is part of Joe Vilgasch's PhD defense. It was a really wonderful big study, and some of the participants are in this room, showing that mindfulness training can reduce activity in the neurologic pain signature, which is a best validated marker so far for pain. And we've been doing this, for all kinds of different conditions. So I put this in here because we're sharing, 20 or 30 different brain models, and they're all open source. So they're all things that work for new individuals, and you can use as targets for different kinds of things like fear, threat, drug craving, reward, pleasure, pain of different kinds. And so I'll leave it there. I won't tell you what we're learning from that, but I think this is telling us more about the architecture of human feeling in the brain. So I'm very grateful to be here. I feel like a lot of love in this room and a lot of, definitely in a very enriched environment. So thank you.
Our next speaker is Dr. Ceci Westbrook, who was a graduate student, MD, PhD from 2012 to 2017, and probably has on record the most awkward conversation with Richie ever, which I encourage you to ask her about.
Thank you so much. So I heard this was supposed to be a little more about us, so that's how I structured my talk a bit. So for those who don't know me, I'm from Chicago. I went to college in Pittsburgh, and I was here for almost 10 years for the MD PhD program, and I was in the lab for 5 of those. And as proof, I did in fact graduate, and I did in fact meet Bucky, which is more important. And here's me at match day as well. And then I was looking for old pictures. I didn't have a lot of time, but this is one picture of some of my closest buds, and then I couldn't find one with Sasha, but we have to take one today or tomorrow before we all go home. So since then, I ended up moving back to Pittsburgh, where I went through residency and child fellowship, and I'm a newly minted clinical attending, so well, that's a lie. I'm a postdoc, but I do get to be a grown-up one day a week. So I do see patients one day a week. I see primarily adolescents. And my big news is my K-23 was actually funded. I did get actual money. I have an account number. And so I have it under good authority that I will be an assistant professor starting in September, TBD. Yeah, there is money coming out of NIMH right now, at least 1K's worth. So for those who know me, you won't be surprised my interests are primarily in the mechanisms involved in what I'll call repetitive negative thought. This includes rumination and worry primarily. I'm interested in understanding how depression and anxiety form during adolescence. And more recently, I've become interested in computational psychiatry. I do actually have some cool data around the explore-exploit balance, which I'm not going to talk about today, but I can talk about it for those who are interested. But for now, I want to talk a little bit about my work in the rumination and worry space. So this is all work I've been doing on a few data sets where what we do is we get people to ruminate and worry in the scanner. So we interview them, we get them to tell us what they actually worry and ruminate about in real life. Then we have them do that in the scanner, and importantly, then we ask them to disengage from those thought processes and over the course of several trials. So each trial they ruminate, and then we try to get them to disengage using different instructed types of strategies. Different types of strategies and disengagement. So on one paper we have published, we did examine mindful disengagement from worry. And what we found is that when people reported more success at disengaging, they had more brain activity in this brain region, which is rosterolateral prefrontal cortex, which Gina also mentioned, which is a region that's really involved in sort of metacognitive awareness, higher order thinking, etc. And so from this, we concluded that this type of thought must be needed for disengagement from worry. However, more recently, I've been working on a data set where we have people disengage implicitly. We basically just ask them to do another task. In this case, it's the SART, which is a very boring go-no-go task. It's a task of sustained attention. And here what we found actually is on trials where people reported more success disengaging, the brain networks that they used were the dorsal attention network, not surprising, it's a sustained attention task, and the salience network, which is important for switching attention. And so from this, actually, what we can conclude is maybe the abstract thought is really just for mindfulness, but that actually what's key is this ability to switch attention using the salience network, meaning that if you want to disengage from worry, it may matter less what you're switching your attention to than the ability that you are able to switch your attention. So that's a theme I'm exploring more. And then this is cool, but the next work I'm really excited about, which is really hot off the press, from the same data set, is using representational similarity analysis to see if we can actually detect reactivation of specific thoughts prior to people making errors on the SART. So this is busy, so I'm going to try to break it down quickly. But what you're seeing here is on each one of these little graphs, this little line is showing the moment that people make an error. And on the top row, they're making errors of omission. On the bottom row, they're making errors of commission. And then these are different templates. These are different RSA templates. So here is looking just at the thought that they just thought on that specific trial. This is an average of all that subject's thoughts. And then this is a between subjects average, which is using more traditional MVPA. And what I want you to notice here is that prior to making an error of omission, we see reactivation of really any kind of thought, so anything specific to that participant. But before they make an error of commission, we only see come back to that. We only see thoughts from that specific trial. So we see activation. I don't know if the mouse will work, but so this is showing that it's really, we see reactivation of the specific thought they were just perseverating about right before they make an error of commission. So I think this is really cool, and we're going to do more with it. But for now, this is my current work in child development. This is my son, Dominic. He's 9 months old, and hopefully you all get to see him tomorrow. He would love to meet all of you. Thank you.
Thank you, Ceci. Would love to meet him. So now I will introduce David Perlman, who was a graduate student from 2006 to 2016. And you are going to go with no slides.
I'm going to go with no slides.
There we go.
Yes. I'm going to compete with Jeff Maxwell for the least academic of anyone here, although he might still win because having a non-academic job is probably less academic than having no job at the moment. Anyway, as Regina said, I was a grad student from 06 to 16, and when I first arrived, Richie asked me to create the first phase of the research project on Tukdam, which was incredibly interesting, and I spent many years of most of my life focusing on that. A beautiful documentary film came out of that, but scientifically speaking, it was a very challenging thing to work on. And what I ended up finding was that there was this huge disconnect between something that was incredibly experientially real to me versus very hard to pin down with physical measurements. And I'm not completely proud to say that my reaction to that was that I had a pretty big spiritual crisis around it, and I kind of ran away, which may not have been the right choice at the time. More on that later. Anyway, I graduated in 2016, and for the purpose of trying to develop a tech career in I went to Silicon Valley, and on Brianna Schuyler's advice, I did a data science project on open source data having to do with social networks and political disinformation, which you might recall 2016, that was 17, that was kind of an interesting time, although they're all interesting times now. Lucky for all of us. The work that I did caught the attention of some interesting people. I met some very interesting military cybersecurity people. I gave talks to Special Operations Command in Tampa, talks in Washington, D.C., Lawrence Livermore National Laboratory. I wrote a book chapter on a book that they put out every year on emerging strategically relevant technologies. I did a full-day workshop for a bunch of military leaders, NATO military leaders in London. It was a very interesting time. And long story short, then the bad guys won, and it all kind of became irrelevant. There was some right-wing conspiracy nuts wrote a report labeling me and my colleagues as the censorship industrial complex, which didn't seem like that big of a problem until it got turned into a congressional report. So I don't really talk to any of those people anymore. It's been a really weird journey, a really weird journey. Anyway, in recent years, I've had, because of my own developmental process, I've stopped and I've reevaluated what do I really think is important, and I've come back around to thinking, how do I resolve that spiritual philosophical crisis that I came to from the Tukdam project? And what I've realized is that as it's been simmering in the background, I feel like I've learned a lot passively, mostly through my own focus on my Tibetan Buddhist practice, without anything else is kind of mixed in with it, for me personally at least. I think that it's an important question. How do you deal with things where it's hard to objectively pin down what's really real, even when there are things that are really real? And I think that this is applicable in a lot of different areas, starting with the questions having to do with mind-body dualism that a couple of the last couple of people were really hinting at in ways that I think are very interesting. So I believe with Richie's blessing, I'm planning to move forward and go back and write some of this up in a way that I hope will be useful to people. When I've spoken about it in the past, it's gotten a positive reaction, so I'm hoping that I can contribute something of value. And as I've been thinking about it, I've realized that there's also even a bit of a through line with the other things that I did, in the sense that these questions of social networks and political disinformation and so on and so forth, it raises some of the same questions of how do you figure out what's real in kind of a new world where the standards of evidence, both in terms of what's actually available to us and in terms of the changing ways that people think and changing structures of the mind even, it's harder to pin down what's real, but I think it is still important to have ways of thinking about what's real that's not just, I'm going to be a hard-nosed reductionist. I'm only going to believe, the most concrete things. And on the other hand, you don't want to fall into the trap of just being kind of a fuzzy-headed woo-woo person who just, believes anything that anyone says, if it's dressed up nicely. So that's where I'm hoping to go in the future. I think I'm well under the 5 minutes, and that's all that I have to say.
Been so delightful, and we have another short break. We'll be back here in 15 minutes at 3:30. I'm looking at you, Chris.
That's perfect. Thank you.
Thank you, everybody.
Where's Nate back?
go put yourself in front of the camera.
Thank you. I'm so glad to be here, and I'm so grateful for people who organize this, because this is really a special moment that I feel like doesn't happen in academia very often, so I'm really just grateful to be part of it. So I'm going to quickly take you through a few things we do in my lab, and then get to the heart of things, which is where I kind of sat with my time in the lab, which was almost 20 years ago, and thought about what Richie taught me. So in my lab, we use functional MRI and either assessment of or administration of hormones to study a variety of disorders, including eating disorders, anorexia, and avoidant restrictive food intake disorder, mood disorders, including sex differences therein, obesity, including treatments, and neuromodulation, and substance use disorders. So I'm going to really quickly go over a smattering of our more recent findings, just to give you a flavor of what we do. So as part of a collaborative at MGB, the Multidisciplinary Eating Disorders Research Collaborative, our findings more recently in eating disorders focus on avoidant restrictive food intake disorder, which, if you're not familiar, kind of entered the DSM within the last 12 or so years. It is typified by 3 phenotypes, which you see here. And so our publication last year was one of the first neuroimaging studies to kind of document the neural mechanisms underlying some of these flavors. I'm not going to go into details. You can kind of see this. You can look up the paper. But that is what we're doing now, is looking at CBT for ARFID and the effects on brain and behavior. In the terms of mood disorders, not surprisingly, my interests also lie in appetite, specifically whether opposing appetitive phenotypes, so hypo and hyperphagic depression, differ in brain and hormone function. So a shout out to my academic sibling, Diego Pizzagalli, who was a collaborator on the grant that led to these data. Postdoc in my lab, Sarah Brassard, recently led some analyses looking at functional connectivity during anticipation and receipt phases of food and monetary reward processing, and documented differences in connectivity between hyper and hypophagic depression. In the domain of obesity, a few years ago, I collaborated with some basic scientists to do a translational study revealing a dedicated role for the cerebellar neurons in the deep lateral cerebellar nucleus to regulate dopamine circuits influencing feeding behavior. I'm not going to go into the details on this, but what it led to is a collaboration with some colleagues who use cerebellar TMS to develop a feasibility study in Prader-Willi syndrome, which is an area I studied before I came to Richie's lab. So it was really nice to kind of get back into this. Cerebellar data, it's working in the brain, it's working in behavior, very exciting. Now we're doing a study in healthy adults, blah, blah, blah. Also, substance use disorders, GLP-1s, looking at efficacy with my collaborator, Georgi Suzuki, and also a psychedelic study we're about to start. Now, this picture maybe a few years ago, but what I want to just really briefly tell you about is what I learned in Richie's lab. I came with a very humble set of skills, and I learned so much. This is just a brief smattering: shell scripting, AFNI boot camp, eye tracking, so many more things that really increased the rigor of my science. I'm so grateful for that. How to present data, really distinct memory of Drew Fox showing activation clusters in white matter, and how did it, like, no matter what you did, you could not get rid of it just sitting in white matter, and what did that do? And the discussion that we had around it and the ideas that people had were so inspiring about how you do science. And it eased my dread a little bit about having to give my presentation, but learned so much about how you present your science. How to lead a lab. Many of you have spoken to the breadth of things covered in Richie's lab, and I've tried to continue that in my own lab. And along with that, I think another thing that stays with me is the fostering of open discourse. I think back in the mid-2000s, we were relatively new in this field, and we wrestled with these ideas of, ancillary findings at an MRI and pregnancy tests in minors, and how do you deal with these ethical issues, and just really how Richie fostered discourse around these kinds of issues was very inspirational. And finally, how to survive as an academic. I applied for a foundation grant and NIH grant while I was in Richie's lab, and he's really demonstrated how you have to work as a scientist to survive in this world. And finally, all I'm gonna add is that not only in my science have I been inspired by Richie, but it turns out that I married a guy from India. We travel in India a lot, and we went to the Isha Yoga Center in Coimbatore in Tamil Nadu. This is my husband and my younger daughter. It was very hot. That's why she looks like that. But we were able, you can't take your cameras in, so that's why this is a photo from the outside, but we were able to sit and meditate, and it was one of the most transformative experiences that I recall in my life. And so I've tried to continue that in my personal life, and I'm just really grateful for all of these things. So those are just acknowledgements, but thank you.
Nobody told me I would have to use a Windows machine.
Neuroplasticity. Hoptman.
You left them all open?
Why don't you work on the comments? You got it?
Unsubscribe. Ugh. There we go. It is my pleasure to introduce Dr. Matt Hoptman, who was a postdoctoral fellow in 1991 through 1993.
Hi, and thank you for having me. It's really a wonderful experience to be here, see so many people who I've known for a very long time, and some people I just met who I'm really glad to have met. And I'd like to just open, before I get into the meat of my talk, and I'll keep it as short as I can, and I shouldn't get the duck, is ways in which Richie's had influences on my career in ways that I knew about and ways that I actually didn't really realize until later. So one was that he took me on as a postdoctoral fellow in the study of healthy aging. I knew nothing about the area except for the fact that I had some hypotheses, and he took me on, and we actually made a good showing of it. We published a few papers on it, and had a very nice paper in Psych Bulletin as well, and it was a very good experience. A second area was a paper that Chris Larson, Richie, and Kathy Putnam wrote about dysfunctional emotion regulation as a prelude to violence, which I had no idea would well, I sort of actually, when I saw it, I sort of went, wait, this might be something interesting. And then the third thing was that I'm currently involved in a R61, R33 randomized controls trial of mindfulness-based virtual reality to reduce impulsive aggression in schizophrenia. So that's a lot of fun and interesting. I'm the site PI, I'm not the main PI. And then the last thing I'd like to say is that Richie really got me to think in a new way about rigor, and rigor in terms of studying individual difference measures and using questionnaires and tasks that are meaningful. So what I'm going to talk about today has to do with some of my work in schizophrenia. So this work has focused primarily on emotion-based impulsivity, which is also known as urgency. And urgency comes in two flavors. So it's been defined as rash action in the context of strong negative emotions. So that's the case, negative urgency, and positive urgency, which has to do with positive emotions. It's measured using the UPS, which was derived from a factor analysis of the 18 self-rated impulsivity questionnaires that were probably the best ones by Whiteside and UW alumnus Don Lynham. And it's elevated in numerous psychiatric disorders, and it's related to many problematic symptom dimensions. So I just listed 2 review articles in which it's covered, one of which is quite recent. It's understudied in schizophrenia. Most of the literature, if I may be so bold, is from me. It has good psychometrics, even in schizophrenia, which many of our patients have very variable response styles. But their coefficient alpha for urgency is at or greater than 0.78, so it's pretty good. So in terms of aggression, so this was work that I completed in 2014 with Dan Javitt, okay, where we showed that urgency was selectively reduced in schizophrenia with effect sizes of greater than 1.2, related to reduced cortical thickness in a number of prefrontal and medial regions, and related to reduced functional connectivity of those regions with other prefrontal and medial prefrontal and ventral frontal regions. And you can sort of see those in the highlighted colors, which are keyed to the regions that you see in the upper right panel, which is where the cortical thickness was measured using FreeSurfer. I then shifted gears to look at urgency and suicidal ideation and behavior in schizophrenia. We found that it was elevated in people with high versus low schizophrenia, suicidal ideation and behavior. There were other measures related. We also showed activation deficits in emotion regulation task in people with high versus low suicidal ideation and behavior and schizophrenia. And importantly, in many of those regions, reduced bold activation in that task was correlated with urgency in multiple emotion regulation regions. And you can see some of those regions on the figure, which I will not go into detail because of time limits. And I will skip this slide as well, given that I only have a minute. But in terms of future directions, I'm currently working on an R01 that's extending this work to depression and in a decent sized sample. Those other studies were smaller. Planning an EMA grant to examine causality among urgency, emotion regulation, and suicidal ideation. And as a shameless self-plug, we had 2 special issues in Frontiers in Psychiatry with QR codes there, one of which is done, the one on the left, the other one I still have to write an editorial for. And I would like to thank all these people, but give a special shout out, of course, to Richie for being a wonderful mentor. So thanks, everyone.
It is my distinct pleasure to invite up to the stage the person who actually hired me into Richie's lab.
But not the first to interview.
Not the first to interview me, for that was Will Irwin, who many remember. But welcome Diego Pizzagalli, who was a postdoctoral research fellow and research scientist from 1999 through 2002.
Yeah, it's wonderful to be here, really. I've not seen some of you such a long time, and of course, I'm so delighted to be here and celebrate Richie. So as you can see, I chose a highly scientific title. But I'll start with the story. So, again, this was 1998, and this was a time where people didn't have, at least we didn't have a computer at home. And so we would go and visit my in-law, and on the way home, we would just stop to the lab to check my email to see whether this distinguished Dr. Richie Davidson might have replied to me in terms of my desire to join his lab. And to this day, I still wonder, why did Richie invite me to his lab? I grew up in this tiny place, beautiful, in Switzerland, not in an academic environment, and for some reason, I just wanted to go to his lab, and as many people in this room, you just open so many doors, and it's not an exaggeration to say that my life, my professional life, and also personal life has been completely transformed by just this experience back then. Yeah, quite a journey from this little village to the mecca of effective neuroscience. So I'm amazed Chris hasn't changed a bit, but I've become a bit gray, but this is pictures that I have from the lab. And this is actually me with our two kids in '99 in our university housing apartment. So I had the brilliant idea of moving the family on December 28. And I remember my wife, we arrived and she almost wanted to go back because it was brutally cold, even coming from Switzerland. But so we were in Madison. And so what I've done after leaving Richie's lab, I spent 23 years in Boston, and basically, a variety of approaches to study depression. Also, I came to your lab. I'm not clinically trained. I had never done work in depression, and you gave me a data set, the infamous PET DAP data that I had heard had burned a couple of postdocs before. I didn't know a clue, so sure. And yeah, that completely changed my trajectory, and I started really becoming extraordinarily passionate about using neuroimaging to study depression. At that time, it was not fashionable to say that the DSM was flawed, but in many ways, you were always ahead of the curve. And something that it drilled in my brain was that depression is just too heterogeneous to study. And so I basically, throughout my career, I've used a variety of approaches for parsing the heterogeneity of depression. And in particular, studying anhedonia, just studying different anhedonic phenotypes, these days also across species. But what I wanted to show you just an example of, now where are we going with this work. We also have done a lot of work looking at trying to identify biomarkers that eventually might be used to guide treatment selection in depression. So in some of the study early on, this was results from the EMBARK study, we basically asked a simple question: can we identify behavioral or neural markers which in this case, for example, ended up predicting response to an atypical antidepressant, bupropion, which has some dopaminergic effects, after failing 8 weeks of treatment with an SSRI sertraline. And what we found was that people that actually had a higher ability to learn from rewards and also had a more functionally coupled brain reward system, so their accumbens talked much more frequently with the other part in the medial prefrontal cortex, that these people actually ended up responding to bupropion after failing 8 weeks of treatment with an SSRI. And based on these and other findings, actually we just published this last week, this just came out, we're now using some of these biomarkers now prospectively, where we are using them to decide how to treat patients. And this was a proof of principle study, but basically we found that people that had some of this biomarker had a 71% response rate versus about 43% among those that did not have these biomarkers. And so, again, as some of you know, after 23 years in Boston, I decided to move my life to California to start a new research institute focused on depression, and these are the type of study that we cannot wait to continue to do. So. You changed my professional and personal life in many ways, and I'll be always grateful for that. And so thank you so much for having me.
Our next speaker needs no introduction, for he has already been introduced. My good friend, collaborator, and constant source of red teaming. For those of you who are there this morning, Dr. Alex Shackman.
I'm Shackman, and it's a real pleasure to be reunited with so many old friends and to have the opportunity to make some new friendships and to renew old ones. And to have this opportunity to co-ruminate on fear, anxiety, and family. So first we got to get in the Wayback Machine and return to the fall of 1994. I was a newly minted psych major. I was eager to join a lab. And so I wandered all over the building, and I was looking for flyers, and I found one on the bulletin board up on the 3rd floor, which is still there. And it looked something maybe a bit like this, some squiggly lines at the top, a colorful cartoon brain, a topo plot, and a headline that read something like, "Are you interested in learning about the brain and emotion?" And Robin Nusslock said something very similar, which was, when I read that, something clicked for me, or started to click into place. Yeah, that is exactly what I think I want to do. And that only increased over time. So I turned on my heel, I plunged through this door, and started talking to this guy, the wonderful Steve Sutton, who ended up hiring me, training me, essentially raising me, mentoring me, getting me cleaned up. And one of the many things Steve did for me is introduce me to this character. So fast forward to the spring of '97, armed with letters from Steve and Richie and Absalom and Craig Barrage, I managed to talk this illustrious crew of investigators at New Haven into an interview for the NTP. An interview where I marched into Mike Davis's office waving a heavily annotated copy of the paper that he had under confidential review at Behavioral Neuroscience. Wonder how I got that. Somehow this brash, eccentric, Shackmanian approach worked, and Yale made me an offer. I was admitted, but I declined after much hand-wringing and picked this guy again. And Richie and I soon embarked on my first-year project focused on, who would have believed it, fear and anxiety. Well, one thing led to another, and before you know it, a decade's passed. It's the summer of '08, and I'm done. I defended my dissertation. I'm still fixated on fear and anxiety, and across that span, I was blessed with so many people who are so important to what I have become, what I am as a person. But all good things must come to an end, and in the spring of '13, we moved our family to Maryland. Here's our comically large orange truck. And I soon got busy down in College Park at the University of Maryland, and along the way, Wisconsin never left our hearts, and my intellectual training never left my mind. We kept on sifting and winnowing. Now, right, as an assistant professor, as folks like Zhao know, I got to do science, I have lab admin, I got papers, apparently a book chapter to write, and among those things, I got to start teaching. Well, guess what? Many of you are guest speakers are in my classroom, undergraduate and graduate today. People like Diego, Tom, Kat, Richie, Ned, Danny and Chris, Decker, Kristen Buss, Av, more Richie, HHDL, more Richie, Brianna, Key and Tor, Ned and Drew, Jerry, Paul, more Drew, too much Drew, Max, Hill, and this one's just for Richie. So outside the classroom at Maryland, I've had the joy of starting my own academic family and starting to make even more good memories with different graduate students and postbacks and staff and administrators, staff scientists, got a new scanner, got tenure, interns, grad students, political advocacy, hiking, birthdays, and one of the real pleasures of the job has been starting to see my own trainees go on to their own faculty careers, like with the Lakra. So just like my academic kids, my biological kids got bigger too, in case you were wondering. Micah graduated high school, Hannah graduated middle school. And one of the genuine blessings, one of the important blessings of the journey that Steve and Richie launched me on back in the 90s has been the opportunity to continue working and thinking and playing with so many old friends, folks like Jonathan, meet Drew's family, hang out with Tal, write books with Regina, go hiking with Manu, who was in Diego's slides, hang out with Rotem and Dan, and drink soda in academic libraries. The list goes on. We get to do stuff with Diego and Ned and Aaron and Chris and Regina, and sometimes even Nate. Now, years ago, Tim Solomon said, I have a pathological need to mentor, and I guess he's right. One of the most satisfying parts of my job has been the opportunity to start to get to know and mentor and collaborate with my friends' kids. People from Drew's lab, from Tor's lab, other folks from Chris Larson's lab, and this is the Masuda Okan of the Plato quote from this morning.
With regard to my science, I remain just as excited about fear and anxiety, multidisciplinary approaches, coordinated cross-species platforms of the kind that Richie, Liz Phelps, and others modeled for me, that Brad Postal modeled for me when I was being trained. So thanks, amygdalot. Is it Travers or Urry?
Thank you, Alex. And now it is a pleasure to introduce Dr. Brittany Travers, who was a postdoc from 2011 to 2014 in Richie's lab. Welcome, Brittany.
It is really awesome to be here with you all today. Thank you so much. So I decided to give kind of what my lab has been up to since, I guess, moving on from the postdoc. So I'm still here at UW-Madison, but in kinesiology and occupational therapy program. And there's three major questions that we've really been trying to tackle. So the first one is, are there sensory motor differences in autism, and do they actually matter? And what we have been finding is that sensory motor differences are actually even more prevalent than what we thought. This is data from the CDC, and we're finding that 72% of autistic children have some type of significant motor difference, and it's 74% with sensory. We also have been showing that motor differences are either sustained or widen into adolescence and adulthood, and we actually have two different data sets that have been able to show this. And we also know that sensory motor features are related to daily living skills and autism across a number of age groups. So we do think that these motor differences are not just kind of a difference, but they might actually be having an impact on daily life. Recent work from our lab suggested that across all the different motor measures that we were looking at, that it was actually the fine motor skills that had the strongest associations with the daily living skills in the autistic children and adolescents. So that's on the behavioral side. The second question we've been really asking is, what are the brain associations with these sensory and motor differences in autism? And in 2015, we had results, and they were in the brainstem. I was like, what is happening, right? It was one of those things that it's like, oh, maybe we should just cut that off. It's really noisy. But we decided to follow the signal and to really try to better understand what's happening. But it took, the last probably 11 years have been kind of dedicated to this, really thinking about how do we collect data images that actually are able to show us the brainstem and its fine small little structures within it. So we have data collection techniques that collaborators and I have been working to develop, as well as post-processing techniques that people like Jose Guerrero Gonzalez and Olivia Sargent, both trainees, have been using to try to even further improve our ability to look at brainstem nuclei in our data sets. So with this in hand, we were awarded an R01 in 2018 to really try to understand the brainstem in light of core autism features and sensory motor features. And there's so much I wanted to show you, but this is what I kind of landed on, where we looked at brainstem pathways and found, particularly with diffusion-weighted imaging, and we found that they were related to sensory features in autistic children, particularly hyporesponsiveness and tactile responsivity. And one of the things that I think is really important about this is that the brainstem and cerebellar white matter tracts were only 7% of our work, or of our search space, but they were about 40% of our findings. And this is an area that we've actually been omitting for so long. I also think it's important that a lot of these brainstem structures may suggest that some of these behaviors are reflexive, so any type of intervention that asks a child to control these is probably not going to be that effective. So we now have moved into the renewal phase of the R01, which has been really fun. I'm getting to work with Nagesh Adluru and others on this, and we've been looking at how do we best come up with tractography with the brainstem and look at how the brainstem's connected to the rest of the brain. We're also very interested in not only the sensory motor associations, but feeding and heart rate variability. So I'm really excited to talk more with Laura and others who are interested in the feeding piece as well. well. The last part is the impact of sensory motor intervention. So Jamie asked me, are you still doing We Balance Board stuff? Well, yes, sort of, right? So you all in the community that you created really gave me so much feedback on this. We were able to do a cross-sectional design and then also a follow-up randomized control trial, and I'll summarize those here. So we did find balance improvements within the balance intervention. We actually also saw changes in the prominence of autistic features. We did not see the daily living skill changes that we had hoped to see in the balance piece, but we did find changes in brain wiring that were actually quite distinct in the autistic group, which is something that we're following up on and trying to understand more. So that is a brief overview. I want to say thank you to Richie. I think that the biggest impact is I was co-mentored, right? But you never made me feel that I was anything but central to the things that you were doing here. And you created a community where I learned from so many of you along the way, and I'm still learning from you today. And for that, I am so grateful and hoping to continue to propagate that in the community and the lab spaces that I'm creating with my students. So thank you.
Thank you, Brittany. That was fantastic. All right, so now I am happy to, well, first see if I can use a Windows machine. Oh, wait, is this the right one? No.
See right there.
Oh, I see. Okay. Is this
Selective
right?
attention. Or search.
But I can't. Is this the right one? Yeah, okay, there we go.
She also uses keynotes, so she's less.
Yeah, even more neuroplasticity happening now. All right, so now it's my pleasure to introduce Dr. Heather Urry, who was a postdoc from 2000 to 2004 and an assistant scientist from 2004 to 2005. Welcome, Heather.
Sorry for that, Richie. Good afternoon, everyone. I'm really can't imagine being anywhere else but here in Madison for this event to celebrate so many years of, well, learning and growing. And I wouldn't be the same person had I not had that experience. Yeah, so I think Regina already gave the history. I was a postdoc in the lab starting in 2000 and became an assistant scientist before I then moved off to Tufts University in 2005. And really looking back, I still can't believe my good fortune. Well, there's a couple of things I'll tell you about, but there were serendipitous events really that led me to Richie's lab. Maybe some of you had also your own serendipitous events. So there are two that I'm going to just draw attention to here. There we go. So the first was ending up in Madison for my internship. So I did my PhD in clinical psychology and was at the UW Hospital and Clinics for the first year. So I was in the right place at the right time. But the right time was the most amazing thing, because what happened was I somehow screwed up the courage to email you, Richie, and I said, I'm here in Madison. I really would love to work with you. I'm so interested in emotion regulation and health and sort of mind-body stuff. And you said, well, that's interesting, because I just got funding for the Mind Body Center, which will centrally feature emotion regulation. And so I just couldn't, like, what are the odds that that would happen? And in fact, the odds were, in my mind, so low that when you eventually emailed me to say that you would indeed be able to offer me a postdoc, I swear to God, I thought to myself, like, did he write the right Heather? There were a lot of Heathers in the world at the time. And I honestly thought maybe it was a mistake, but you didn't have apparently the heart to tell me that was not the case. And I really have never looked back. I also would take an entire 5 minutes really to thank and recognize all the people that contributed to my training aside from Richie. I won't do that because we just don't have time, but I do want to recognize two people who were, in addition to Richie, central to my development, and that is Carien van Reekum, who is here in this room, of course, and Tom Johnstone. And together we spent a lot of time thinking about emotion regulation, and especially how do we use the way we think about the things that we experience to alter how we're feeling. And in the course of those studies, there were a few really interesting and I think important findings that sort of led me along the way to the kind of research that I did at Tufts. The first was this finding by Tom and colleagues showing that when we ask people to use reappraisal, that we have, really, 2 minutes? how does that happen? But anyway, yeah. Okay. So the first was like the finding that when we ask people to use reappraisal, that they actually spend a small but statistically significant smaller amount of time actually with their eyes open when they're looking at pictures. So that seems potentially problematic. And then combined with Carien's finding, which was based on imaging data where we also collected eye tracking and found indeed that when we ask people to use reappraisal to decrease their emotional responses, that's in the red bar there, that in fact people are spending less time looking at the pictures. And so, I started to really be concerned about this because we were making claims about what was the impact of reappraisal, right? Actually altering your thinking, and yet here was evidence that maybe it's more about where you're looking. So I started to think, well, barring unethical, sort of barbaric-looking machines like this, how are we going to actually prevent people from looking away from the images so that we could actually see how they're thinking and whether or not that would have an impact? So I ended up developing what's called the gaze-directed cognitive reappraisal task, and I won't go into the details for lack of time, but really what we tried to do was get people to think differently about the pictures at the same time that we recorded various kinds of dependent variables and also manipulated their gaze, so asked them to focus on certain areas of the picture so that we would control where they were looking and therefore hopefully prevent that from looking time from being a confound in our inferences. And this has proven to be a useful task. I won't talk about the details, although this is a very cool graph. This is corrugator muscle activity, showing basically that you can separate these two things, that where people look and how they're thinking actually both do alter the way that people respond emotionally. We've also used this task to consider age differences, which are interesting because older people tend to show different looking patterns, and found that indeed changes in the brain do still happen even when we use gaze-directed reappraisal, and that older adults in fact show a pattern that is consistent with lesser ventrolateral prefrontal activation. So I'm running out of time. Just two things. I'm really still interested in these ideas about how to separate out and think about the impact of reappraisal versus other emotion regulation strategies like attentional deployment. Yuan and colleagues really published an interesting paper in the context of depression to be thinking about what are the brain regions that might be responsible for a pathological attractor state that contributes to depression. So that's kind of where my mind is now, but I'm going to need money. And so part of that will require seeking NIH funding, which should be an adventure. And also, really just to wrap this up, because I know I'm out of time, I have spent a lot of time also thinking about, in bigger picture ways, about stress and how that affects performance under stress. So you can ask me about that at another time. I will stop just to say that these are the people who made that possible. And really, Richie, thank you so much for not having the heart to tell me. You meant to email another Heather, so thank you.
So we have something special for Richie. Come on up. I don't know the details. I just know this is happening. Surprise.
In one of the early conversation of the Richie told that his son is the Dalai Lama challenged him that applying or putting the compassion or the mental health in the science or the science research. Throughout this journey of 50 years, he applied these compassion and inner or the emotional hygiene in the science, not just applying, he also applying himself and sharing to all the world. So as I am like a monk researcher and the follower and the student of His Holiness the Dalai Lama, I want to give a white scarf as the gratitude and appreciation to the Richie. Thank you.
Now it is time for Richie's closing remarks, and for those of you wondering, the duck's not off.
Whew. It's really, I'm overwhelmed. I really am. First, I want to just express my really profound gratitude. And I'm so incredibly proud of each and every one of you. To see how you've taken this work and what you're doing with it now is really breathtaking. And I'm really so inspired and moved and also hopeful for the future. And I know each of you is affecting a whole sphere of your own students. And through this work, it is, I think, our collective aspiration that we really can help relieve some of the suffering that is so prevalent in today's world. And that's really our motivation for doing the work that we're doing. And so I'm really encouraged to see all that is being done. There are a few themes that you've all covered that I want to just sort of underscore. One is that every one of you is rigorous, and even tackling really complicated, problematic, complex issues, each one of you has embodied this kind of rigor, and that is kind of essential for making progress, and I so appreciate that. The second is that you guys are not method bound. Each of you has some unbelievable methodological skills, but one of the things that I love is that none of you are bound to any single method. Your methods are driven by the questions you're asking. And this is something that I remember early on in my career, someone came to me and said that they're an fMRI researcher. And I said, what does that mean? I think each of you embody that kind of quality, which I think is so incredibly important. I also really appreciate that all of you in your own ways, and this is something that I would really encourage you to keep doing, be bold and don't be afraid of asking big questions. This is something we really need, and one of you quoted a famous quote that I often say, and actually it's not my quote, so I want to just remind you this was first said by Donald Hebb in, I think it was 1956, and that is, if it's not worth doing, it's not worth doing well. And so it's really important to ask those big questions. Another theme that
I think is exemplified in a lot of this work is something that some of you called out explicitly, and that is the theme of individual differences. This is something that those of you who know me well, you know I've harped on this throughout my career. And I think it was Carien that put it beautifully. I mean, Carien was trained as a social psychologist, and social psychologists are basically taught to ignore individual differences, and they're considered noise. And yet, we come back to this time and time again, and I think that one of the central themes across almost all of these presentations is how important individual differences are. And when it comes to interventions, one of the things that I often say is one size does not fit all. And it's our responsibility as scientists to really try to figure this out and to make better predictions about which people will benefit from which kinds of interventions most effectively. Another important theme is that intervention research can and really should be mechanistic. We can learn about mechanism through doing intervention work. And so I love that many of you who are doing real basic work are also doing intervention work. And you can discover fundamental truths through intervention work. And at the same time, you can be of direct benefit to people. So I think that's something that needs to really be celebrated and reinforced. Another theme is connections between the mind, brain, and body. A lot of affective and cognitive neuroscience, I would say more cognitive neuroscience than affective neuroscience, is exclusively brain-centric and doesn't so much include the body. And we are embodied creatures. The body plays an incredibly important role in our emotions and our experience, and we need to take account of the body in what's happening. And Melissa's work is a great example of really deeply diving into understanding brain-body relations, mind-brain-body relations that are really incredibly important. We heard about measures, many different kinds of measures, and various comments, jokes, and serious analyses were presented about self-report measures, their limitations, and also behavioral measures. And one of the challenges, I think, in the future that we all need to take seriously is getting better measures of actual experience. And I don't claim to have any answers here, but I'll just mention there's a paper which was published in Nature Human Behavior not long ago looking at the relationship between early childhood abuse and later adult outcomes. This was a paper by Kathy Whittam and her colleagues. And this was a longitudinal study where they had objective measures, objective records of actual early childhood abuse that came from the judicial system. And they also had participants' subjective experiences as they described them in clinical interviews that were coded. And they looked at the relationship between the objective indicators of abuse and subsequent adult outcomes, and the descriptions of experience that were provided by the participants in these interviews, and basically what the finding from this study is. Now, there are other data in the literature that are not consistent with this, so so we need to be careful here. But what this study showed is that it was the experience that was a better predictor of later adult outcomes than the court record findings of the actual abuse history. And so I'm not suggesting that this is the be-all and end-all study, but it underscores the important role of experience that we somehow need to take account of in addition to our measures of behavior and biology. Another theme that was exemplified in some of this work, which, you know, we take this so much for granted, but yet I think it's something that is important to just call out, and that is that there is just a wealth of evidence to show that when we do various things to transform the mind, the brain actually changes. And that is important to just recognize and call out, and that is actually a fact that can be motivating for and very helpful in presenting certain kinds of strategies and interventions to people to help them better engage with whatever it is that's being offered to help them transform their mind. And then I'm not going to talk much longer. Let me just end by saying that there was a kind of magic, and still is a kind of magic, that can occur in a scientific environment where collaboration and interdisciplinarity are so strongly encouraged. One of the speakers talked about fearlessness, and no single scientist will ever acquire all of the skills necessary to answer the kind of complex questions that we're asking. And so what we need to do is to collaborate. And collaboration has been really at the core of so much of the work that has gone on in my career. And you all have exemplified that in so many different ways. And to collaborate in an environment where we can actually be incredibly rigorous and also be cooperative and kind to one another is a challenge in the current academic environment. We're living, and it's much worse, I think, now than it was 20 years ago, simply because of increased scarcity of resources. And so I would encourage all of you, and I know that you do in your own ways, to figure out how you can organize your labs in ways where you can maintain respect, kindness, and rigor at the same time, all embedded within an ethical framework. And this morning there was a beautiful session that Alex and Drew led on contemporary uses of AI in research that was really eye-opening and incredibly provocative. And one of the foundation pieces of that was really the ethical framing of science in a really beautiful way. And this is just a plug to maintain that and do it in a way where we can be also kind and respectful and still be rigorous. So I want to just, again, express my deepest bow of gratitude, as I do often say, which I'm looking at Roshi now, and her deep bows over the course of her life have really profoundly influenced my deep bows. And I just want to end by asking each of you to reflect on how your science really could be of most benefit to others, because that really is why we're doing the work we're doing. And we are all in an incredibly privileged position to have the kind of resources that we have to do the work we have, and let's do it for the greater good. So thank you, and I look forward to celebrating more this evening.